Hello students, I am Mrs. Asha Bansal, a science educator with over 40 years of teaching experience and a former CBSE Evaluator. Welcome to your ultimate one-shot revision for Class 12 Biology. We are about to embark on a comprehensive journey through the entire syllabus. We will cover the most critical exam-centric questions from every chapter that have been repeated in the board exams year after year. So, dear children, have your notebook ready and let us together work to build your confidence slowly, one by one. Let us begin with the first lesson, sexual reproduction in flowering plants. The important question from this lesson is explain the process of double fertilization, describe the journey of the pollen tube and the fate of the two male gametes. Why this process is unique to the flowering plants. Let us understand double fertilization. When a pollen grain lands on a compatible stigma, it germinates. The pollen tube carrying two male gametes grows through the style and enters the ovule through a very small opening called micropyle. Once inside the embryo sac, the tip of the pollen tube ruptures, releasing the two male gametes. One male gamete fuses with the egg cell. This fusion is called syngamy and it forms the diploid zygote which will eventually develop into the embryo. The other male gamete fuses with the two polar nuclei present in the central cell. This fusion of three haploid nuclei is called triple fusion, resulting in a triploid primary endosperm nucleus called PEN, which develops into the endosperm, a nutritive tissue for the embryo. Since two fusion events, syngamy and triple fusion occur, this process is called double fertilization. It is unique to the angiosperms, the flowering plants, and is their most significant characteristic, ensuring that endosperm develops only after fertilization, saving the plant's energy. Next question, differentiate between microsporogenesis and megasporogenesis. This question tests your understanding of gamete formation. So first let us understand microsporogenesis, that is the formation of the microspore or the pollen grains from the microspore mother cell MMC. Each MMC in the anther undergoes meiosis to produce 4 haploid microspores. Let us understand what pollination is and the differences between self-pollination and cross-pollination. Pollination is the transfer of the pollen grains from the anther to the stigma of a flower. Self-pollination is when the pollen grains are transferred from the anther to the stigma of either the same flower or some other flower but of the same plant. While in cross-pollination, the pollen grains are transferred to the flower of some other plant but of the same species. Certain agents are required for self-pollination. They could be wind, water, that is abiotic. For cross-fertilization, it could be external agents which could be biotic or abiotic. Now, the offsprings produced for self-pollination, they produce genetically similar because same gametes are combining. While in cross-pollination, they produce genetically diverse offsprings. That means the variations occur. Some examples of self-pollination: pea, groundnut. For cross-pollination, the examples are apple, maize. Let us begin with the next lesson, human reproduction. Question 1. Draw a label diagram of the human male reproductive system explain the structure and functions of the testis, epididymis, and accessory glands, and semen formation. Let us look at the male reproductive system. It consists of paired testes, accessory ducts, accessory glands, and the external genitalia penis. Structure and function of testes. The testes are the primary male reproductive organs. They are located outside the body, outside the abdominal cavity, within a sac called scrotum. Why are they outside the body? Because spermatogenesis requires a temperature 2 to 3 degrees Celsius lower than the normal body temperature. Each testis is composed of about 250 compartments called testicular lobules. Each lobule contains 1 to 3 highly coiled seminiferous tubules. Inside these tubules, two main types of cells are present. The first one is the male germ cell spermatogonia. These cells undergo meiotic division to form sperms. The second type of cells are the Sertoli cells or the nurse cells. These provide nourishment to the developing nerve cell. Between the seminiferous tubules, there are Leydig cells or interstitial cells. Their function is to secrete the male hormone testosterone. Now, the structure and function of epididymis. The epididymis is a long coiled tube that lies along the border of each testis. It has three parts: the caput or the head, corpus or the body, and cauda or the tail. Its functions are storage. It acts as a temporary storage site for spermatozoa. It is a site for maturation, means sperm gains motility and fertilizing capacity as they pass through the epididymis. It also acts as transport system because it transports the sperm from the testis to the vasa deferens. Now, the accessory glands and semen formation. The male accessory glands include a pair of seminal vesicles, a single prostate gland, and a pair of bulbourethral glands or the Cowper's glands. Their secretion, along with the sperm from the testis, constitute the semen or the seminal plasma. Seminal vesicles. They secrete an alkaline viscous fluid that constitutes about 60 to 70% of the semen volume. This fluid is rich in fructose, which provides energy to the sperms, prostaglandins, and clotting proteins. Prostate gland. It secretes a thin, milky, alkaline fluid that activates the sperm and helps neutralize the acidity of the vaginal tract, protecting the sperms. Its secretion contains calcium and certain enzymes. Bulbourethral glands. They secrete a small amount of clear, slippery fluid before ejaculation. This fluid lubricates the urethra and neutralizes any residual acidity from urine, preparing a safe passage for the sperm. Together, these fluids provide a suitable medium for sperm survival, motility, and transport. Describe the process of oogenesis in human. How is it different from spermatogenesis? Mention at least three differences. Now let us understand the formation of the female gamete, a process called oogenesis. It is the process of formation and maturation of the ovum in the ovaries. Unlike spermatogenesis, which is a continuous process, oogenesis is discontinuous process. Remember that. Phases of oogenesis. It occurs in three phases. The first phase is multiplication phase. This phase begins during fetal development. The germinal epithelial cells of the ovary divide by mitosis to produce millions of diploid cells called oogonia. No more oogonia are formed or added after birth. This is important. Please remember. Second phase is growth phase. Each oogonium grows into a larger diploid cell called the primary oocyte. Each primary oocyte gets surrounded by a layer of granulosa cells forming the primordial follicle. The primary oocyte enters the first meiotic division, prophase 1, and gets arrested at this stage. It remains in the arrested state until puberty. A girl is born with all the primary oocytes, about 2 to 4 million, already formed in her ovaries. Third phase is the maturation phase. At puberty, under the influence of the hormones, the primary oocyte completes its first meiotic division. This division is unequal, producing one large haploid cell called the secondary oocyte and a small non-functional haploid cell called the first polar body. The secondary oocyte then begins the second meiotic division, but it gets arrested at metaphase 2. It is at this stage that the secondary oocyte is released from the ovary during ovulation. If fertilization occurs by a sperm, the secondary oocyte completes meiosis 2. This is again an unequal division, producing a mature haploid ovum and a second polar body. The fertilization does not occur, the second oocyte degenerates within 24 hours. Now let us understand how it differs from spermatogenesis. Let us consider the products first. In spermatogenesis, from one primary spermatocyte, 4 functional motile sperms are produced. And in oogenesis, from one primary oocyte, only one functional ovum and 3 polar bodies are produced. The unequal division ensures the ovum gets all the cytoplasm, and let us understand the continuity of the two. For sperms, it is a continuous process that begins at puberty and continues throughout a male's life. And for oogenesis, it is a discontinuous process. It begins in the fetal stage, is arrested, and is completed only after fertilization, or it degenerates during the reproductive years. The next we will understand release of gamete. The final product in spermatogenesis, that is the sperm, is released from the body during ejaculation. And for oogenesis, the secondary oocyte arrested at metaphase 2 is released from the ovary. The completion of meiosis occurs only if sperm penetrates it. Question 3. Explain the hormonal control of the menstrual cycle. What changes occur in the uterine wall if fertilization does not occur? Draw hormone level graph. The menstrual cycle is a series of cyclic changes that occur in the reproductive tract of human female from puberty to menopause, with an average duration of 28 days. It is regulated by a complex interplay of hormones from the hypothalamus, pituitary, and ovaries. The cycle has two simultaneous components: the ovarian cycle, change in the ovary, and the menstrual cycle, change in the uterus. Hormonal control and phases of the cycle. First is the menstrual phase, which begins from day 1 and continues till day 5. If fertilization does not occur, the corpus luteum degenerates. This causes a sharp drop in the levels of progesterone and estrogen. Then the uterine changes with no hormonal support. The thick vascular endometrial lining of the uterus breaks down and is shed along with the blood and mucus. This is called menstruation. And the third is pituitary hormones. Low estrogen levels stimulate the pituitary to start secreting FSH, that is follicle stimulating hormone. Second phase is the follicular phase or proliferative phase, which begins from day 5 to day 13 it remains. Ovarian changes during this phase is FSH stimulates the growth of the ovarian follicles. These developing follicles secrete estrogen. Because of the rising levels of the estrogen, the uterine endometrium thickens and regenerates. The endometrial cells multiply and the uterine glands increase. That is why it is called the proliferative phase. Then occurs the hormone peak. Estrogen level reach their peak by the end of this phase. Ovulation, which occurs on day 14. The high level of estrogen triggers a positive feedback on the pituitary, causing a sudden surge in LH, luteinizing hormone. It results in the rupture of mature Graafican follicle, releasing the secondary oocyte. This process is called ovulation. And the fourth phase, that is the luteal phase, or it is also called secretory phase, which is from day 15 to day 28. The ovarian changes which take place during this phases under the influence of LH, the ruptured follicle transforms into a temporary endocrine gland called the corpus luteum. The corpus luteum secretes large amounts of progesterone and some estrogen. Uterine changes also take place during this phase. Progesterone prepares the uterus for implantation. It causes the endometrium to become even thicker, more vascular, and its glands become coiled and secrete nutrients to nourish a possible embryo. This is why it is called the secretory phase. What happens if fertilization does not occur? The corpus luteum has a fixed lifespan of about 10 to 12 days. If pregnancy does not occur, it degenerates into corpus albicans. This leads to a rapid fall in the progesterone and estrogen levels. Without these hormones, the endometrial lining can no longer be maintained. It breaks down, leading to menstruation, and a new cycle begins. Chapter 3, Reproductive Health. Moving on to reproductive health. This chapter is not just biology, it's about social awareness, safe practices, and medical solutions that lead to a healthy society. Question 1. Define reproductive health. What is the Reproductive and Child Healthcare Program RCH? Mention three objectives. According to the World Health Organization WHO, reproductive health is defined as a state of complete physical, mental, and social well being, and not merely the absence of disease or infirmity in all matters relating to the reproductive system and to its functions and processes. It implies that people have the ability to reproduce, regulate their fertility, and practice and enjoy safe sexual relationships. Reproductive and Child Health Care Program RCH. India was the first countries in the world to initiate family planning program at the national level in 1951. Over time, this program expanded into a comprehensive reproductive and child health care program, RCH. The three main objectives of this program are to create awareness among people about various aspects of reproduction, including sexually transmitted diseases, STDs, available contraceptive methods, and safe hygiene practices, to provide facilities, support for family planning, and to address reproductive health issues like menstrual irregularities, infertility, and problems related to pregnancy and childbirth, to provide maternal and child health services such as ensuring safe pregnancy and delivery, providing postnatal care, and implementing immunization programs for children to reduce infant and maternal mortality rates. Question 2. Describe the different methods of contraception. Differentiate between barrier, hormonal, and surgical methods. Which method prevents STDs and why? Contraceptive methods are essential for family planning and preventing unwanted pregnancies. They can be broadly classified into several categories. Let us differentiate between the three main types you need to know. Which method prevents STDs and why? Barrier methods, particularly condoms, are the only ones that protect against the sexually transmitted diseases STD. Now the question arises, why? They create a physical barrier that prevents the direct exchange of body fluids, that is semen, vaginal secretion, and blood, which is the primary mode of transmission for pathogens causing STDs like HIV, gonorrhea, syphilis, etc. Hormonal and surgical methods only prevent pregnancy and often no protection against infection. Explain any one assisted reproductive technology ART such as IVF-ET, ZIFT, or IUT. How does it help infertile couples? Infertility is the inability to conceive even after 2 years of unprotected sexual contact. For couples facing infertility, medical sciences offers several solutions known as assisted reproductive technologies, ARTs. These techniques help us bringing the sperm and egg together in a controlled environment. Let us discuss the most well-known technique, IVF-ET, or in vitro fertilization and embryo transfer. Babies born through this technique are sometimes called test tube babies. Now let us learn the procedure of IVF-ET. First of all, ovarian stimulation is done. The woman is given hormones to stimulate her ovaries to produce multiple eggs, oocytes, in a single cycle instead of just one. Egg retrieval, the mature eggs are collected from the ovaries using a minor surgical procedure guided by the ultrasound. Fertilization is done in vitro, that is, the collected eggs are fertilized with the thousands or a donor sperm in a petri dish which contains the culture medium in a laboratory. The term in vitro means within the glass, referring to the process outside the body. Embryo culture. The fertilized egg, now a zygote, divides to form an embryo, usually 2 to 8 cell stage, or we call it blastocyst. Embryo transfer. The healthy embryo is then transferred into the woman's uterus where it implants and develops naturally into a fetus. How it helps? In vitro fertilization, embryo transfer helps infertile couples in several specific situations, like when the woman's fallopian tubes are blocked or damaged, preventing the egg from meeting the sperm, or in the case of low sperm count or poor sperm motility in the male partner, when other fertility treatments have failed. Some other ART techniques, one is ZIFT, that is zygote intrafallopian transfer. The zygote or the fertilized egg is transferred into the fallopian tube instead of the uterus. Other is intrauterine transfer or IUT. The embryo up to 8-celled stage is transferred into the uterus. Third is intracytoplasmic sperm injection, ICSI. A single sperm is directly injected into an egg to achieve fertilization, often used in severe male infertility. Then gamete intrafallopian transfer, that is GIFT. Unfertilized eggs and sperm are transferred together into the fallopian tube, allowing fertilization to occur inside the healthy body. Chapter 4, Principles of Inheritance and Variation. Question 1. State and explain Mendel's law of segregation with a monohybrid cross. Show how the 3:1 phenotype ratio and 1:2:1 genotypic ratio are obtained in F2 generation. The foundation of genetics rests on this law. The law of segregation states that every individual possesses a pair of alleles for a particular trait, and during gamete formation, the two alleles separate, that is called they segregate, so that each gamete receives only one allele. Let us take Mendel's classic monohybrid cross, a pure tall capital T plant crossed with a pure dwarf small t plant. The parent generation, we are crossing capital T tall with a small t dwarf. Gametes capital T from one parent and small t from the other parent. The F1 generation is all tall because they are heterozygous capital T small t, because capital T is dominant over the small t dwarf. So all the plants obtained were tall. Now, in the next generation, when they are self-pollinated, that is capital T small t is crossed with capital T small t, the alleles segregate during the gamete formation. Each F1 plant produces two types of gametes, that is capital T and small t. The F2 generation is obtained by a Punnett square. We get a phenotypic ratio of 3 tall is to 1 dwarf, and the genotypic ratio will be 1 homozygous dominant capital T, 2 heterozygous dominant, that is capital T small t, and 1 homozygous recessive, that is small t. Question 2. What is polygenic inheritance? Explain inheritance of human skin color. How does it differ from Mendel's inheritance? Polygenic inheritance is when a single trait is controlled by two or more sets of genes, that is multiple genes. Each gene has a small additive effect on the trait. The inheritance of human skin color is a classical example of polygenic inheritance. It is controlled by three genes, capital A, capital B, and capital C, located at different loci. The dominant alleles, that is capital A, capital B, and capital C, contribute melanin, while recessive alleles, small a, small b, and small c, do not. A person with genotype all capitals A, B, B, C, C will have very dark skin because the person has all the dominant alleles. A person with genotype A, B, B, C, C, all in lowercase, that is all recessive, will have very light skin because it does not have any dominant allele. A cross between two individuals with intermediate skin color, that is capital A, capital B, small b, and capital C, small c, will produce a wide range of phenotypes from very light to very dark, forming a continuous variation. It differs from Mendelian inheritance. Mendelian traits like short or tall are controlled by a single gene and produce discontinuous, distinct phenotypes. Polygenic traits are controlled by multiple genes and produce a continuous range of phenotypes. Question 3, a very important and often asked question. Explain sex determination in humans with a genetic cross. Who is responsible for the sex of the child? In humans, sex is determined by the type of sex chromosome inherited. Females have two X chromosomes, capital X and capital X, and males have one X and one Y chromosome as their sex chromosome. The mother is XX, so all her eggs carry one X chromosomes. The male is XY, so his sperms are of two types. 50% will carry the X chromosome and the rest of the 50% will have Y chromosome. So when a genetic cross is done, if an X carrying sperm fertilizes an X carrying egg, the offspring born will be a female. But if a Y carrying sperm fertilizes X carrying egg, the offspring will be a male. Therefore, the sex of the child is determined by the type of the sperm, X or Y, from the male. So, the male is responsible for the sex of the child, not the female. Chapter 5, Molecular Basis of Inheritance. Question 1, describe semi-conservative DNA replication. Explain Michelson and Stahl experiment. DNA replication is the process of producing two identical copies from one original DNA molecule. It is semi-conservative, meaning that when a new DNA molecule is synthesized, one strand comes from the original parental DNA and the other strand is newly synthesized. The Michelson and Stahl experiment in 1958 proved this. They grew Escherichia coli bacteria in a medium containing heavy nitrogen N15 for several generations, so all DNA was heavy. They then transferred the bacteria to a light nitrogen N14 medium. After one generation, the DNA was extracted and analyzed using density gradient centrifugation. The DNA had an intermediate density hybrid, ruling out the conservative method. After two generations, they found two bands, one of intermediate density and one of light density. This perfectly matched the prediction of the semi-conservative model, confirming that replication is semi-conservative. Question 2. Explain Hershey's and Chase experiment proving DNA as genetic material. Why they used P32 and S35. Hershey and Chase in 1952 provided proof that DNA is the genetic material. They worked with bacteriophage, that is, the viruses that infect bacteria, which are composed only of DNA and protein. They grew two batches of phages. One batch with radioactive phosphorus P32, which labels DNA because phosphorus is a component of DNA but not proteins. Another batch with radioactive sulfur S35, which labels protein because sulfur is present in amino acids like cysteine and methionine but not in DNA. They allowed each batch of phages to infect separate culture of E. coli. After infection, they use a blender to separate the phage coats from the bacterial cells and then centrifuge the mixture. What result did they get? In the sample infected with P32 labeled phages, the radioactivity was found inside the bacterial cells. In the sample infected with S35 labeled phages, the radioactivity was found in the supernatant, that is, with the phage coats. The conclusion drawn was, since only the DNA labeled with 32P entered the bacteria and directed the production of the new phase, DNA is the genetic material, not the protein. Explain the Lac-Operon model. How does it switch on and off? The Lac-Operon is E. coli is a classical model for gene regulation. It is a set of genes responsible for the metabolism of lactose. It functions like a switch. The operon consists of structural genes, that is LAC-Z, LAC-Y, and LAC-A. They code for enzymes beta-galactosidase, permeases, and trans-acetylase needed for lactose breakdown. They have a promoter gene, the site where RNA polymerase binds to initiate transcription. They have operator gene, a segment of DNA that acts as a switch, and regulatory gene located between. It codes for the repressor protein. How it switches off. No lactose, the repressor protein is active and binds to the operator. This physically blocks RNA polymerase from moving along the DNA, so transcription of the structural gene is switched off. How it is switched on? This is done in the presence of lactose. Lactose is converted into allolactose. It acts as an inducer. It binds to the repressor protein, changing its shape. The repressor is now inactive and falls off the operator. RNA polymerase can now bind to the promoter and transcribe the genes. The operon is in the on condition now. The glucose effect. E. coli prefers glucose. If both glucose and lactose are present, the operon is not fully active. Low glucose level leads to high CAMP. High CAMP binds to CAP, and this complex binds to the promoter, greatly enhancing RNA polymerase ability to transcribe. This ensures the operon is most active only when lactose is present and the glucose is absent. Chapter 6, Evolution. Describe the Miller-Urey experiment. What was its significance in explaining chemical evolution? Let's begin with the origin of life itself. For a long time, scientists wondered how the first organic molecules the building blocks of life appeared on Earth. In 1953, two scientists, Stanley Miller and Harold Urey, designed a brilliant experiment to test the operant Haldane theory, which proposed that life arose from simple inorganic molecules under the conditions of the early Earth. Look at this diagram carefully. Miller and Urey set up a closed apparatus to simulate the conditions they think existed on primitive Earth. First of all, atmosphere simulation. They filled a large flask with a mixture of gases that they believed composed the early Earth's atmosphere, that is, methane, ammonia, hydrogen, and water vapor. Please keep in mind that there was no oxygen in this mixture because early Earth was a reducing atmosphere. Now, the energy source, they continuously passed electric sparks through the gaseous mixture using electrodes. These sparks simulated lighting, which was a major energy source on the primitive Earth. Next is the ocean simulation. Below the spark chamber, they had a flask containing boiling water, which produced water vapor that circulated through the system. This represented the primitive ocean. Next comes condensation. A condenser cooled the gases, causing the vapors to condense and trickle back into the ocean flask, carrying any newly formed compounds with it. This created a continuous cycle mimicking the natural water cycle. They ran this experiment continuously for a week. When they analyzed the liquid that collected in the trap, they made a groundbreaking discovery. The clear water had turned into a brownish, tarry substance containing amino acids, specifically simple amino acids like glycine and alanine. These are the same amino acids that are the building blocks of proteins, and they had formed spontaneously from inorganic precursors, that is, methane, ammonia, hydrogen, and water, simply by adding energy in the form of electric sparks. Significance of the experiment. This experiment was revolutionary for several reasons. First evidence, it provided the first experimental evidence that organic molecules necessary for life could be synthesized abiotically, that is, without the involvement of the living organisms. Then, support for chemical evolution. It strongly supported the theory of chemical evolution, which proposes that complex organic molecules could have formed from simple inorganic molecules on Earth, creating a primordial soup from which the first life forms eventually emerged. And thirdly, foundation for later research. It opened up an entire new field of research called prebiotic chemistry. Later experiments using slightly different gas mixtures succeeded in forming all the 20 amino acids, sugars, lipids, and even the nitrogenous bases found in the DNA and RNA. In essence, the Miller and Urey experiment showed that the basic building blocks of life are not a divine mystery, but a natural consequence of the physical and chemical conditions that existed on the early Earth. Question 2. State the Darwin's theory of natural selection. Explain evolution of antibiotic resistance using this theory. Now let's discuss the mechanism that drives evolution. Charles Darwin, after years of observation, proposed the theory of natural selection. The theory can be summarized in 4 key points. First point, overproduction. Organisms produce more offsprings than can possibly survive. Variations. individuals in a population have a natural variations in their traits. Then comes struggle for existence. Due to limited resources, there is a constant struggle for survival. And finally, survival of the fittest. Individuals with variations that are better suited to their environment, that is, the adaptive traits, are more likely to survive, reproduce, and pass on those advantageous traits to their offspring. Over many generations, this leads to a change in the characteristics of the entire population. The evolution of antibiotic resistance in bacteria is one of the best and most important real world example of natural selection in action. Variation, the mutant. In any large population of bacteria, there is natural genetic variation. Due to random mutations, a very small number of bacteria might possess a gene that makes them resistant to a specific antibiotic. They are the red dots in our animation. Struggle for existence, the antibiotic attack. When a patient takes an antibiotic to treat a bacterial infection, this creates a powerful selective pressure. The antibiotic is the struggle for the bacteria must face. Then survival of the fittest, the selection. The non-essential bacteria, the majority are killed by the antibiotic. They are not fit for this environment. However, the few resistant bacteria survive. They are the fittest in this specific environment containing the antibiotic. Then inheritance and evolution. These surviving resistant bacteria have now less competition for resources. They reproduce rapidly, passing their resistant gene to all their offsprings. Over the time, the entire population evolves to become highly resistant to that antibiotic. The population has changed, it has evolved. Chapter 7, Human Health and Diseases. Differentiate between innate and acquired immunity. Mention any two innate immunity barriers. Immunity is our body's defense system. It is broadly of two types, innate immunity and acquired immunity. Innate immunity is non-specific, whereas acquired immunity is specific. Innate immunity is present in our body from birth, whereas acquired develops during a person's lifetime. Innate immunity is non-specific. It defends against all types of pathogens in a similar way. Acquired immunity is specific. It targets a specific pathogen. No memory, it does not remember a previous encounter, whereas it has memory, it responds more on strongly on second encounter. Innate immunity is immediate, first line of defense. Acquired immunity is slow but powerful and sustained. Two barriers of innate immunity. First is the physical barriers. The skin, it prevents the microbial entry. Mucous membranes, they trap microbes in respiratory or the gut tract. Physiological barriers like acid in the stomach, they kill the microbes. Lysosome in the tears, they break down bacterial cell walls. Second question, name the causative agents, symptoms and transmission modes of any two, typhoid, pneumonia, common cold. Let us quickly review some important diseases. Typhoid. The causative agent is the bacterium Salmonella typhi. The symptoms are sustained high fever, 39 to 40 degrees Celsius, weakness, stomach pain, headache, and loss of appetite. Intestinal perforation can occur in severe cases. It is detected by the Vidal test, and the mode of transmission is contaminated food and water, or you can say fecal oral route. The next disease is pneumonia. Causative agent is Bacterium Streptococcus pneumoniae. It can also be viral or fungal. The symptoms are fever, chills, cough, headache. Severe cases show accumulation of fluid in the alveoli, leading to severe respiratory problems. Mode of transpiration, Mode of transmission: inhaling droplets, aerosols from an infected person's cough or sneeze, or sharing the utensils. Third disease, the most common one, common cold. Causative agent is virus, most commonly it is rhinovirus. Symptoms: nasal congestion, discharge, sore throat, cough, headache, and mild fever. It affects the nose and respiratory passages but not the lungs. Now, the mode of transmission: inhaling droplets from an infected person or touching contaminated surfaces. Explain the life cycle of Plasmodium. Name the vector. Where is malaria difficult to eradicate? Malaria is caused by the protozoan parasite Plasmodium vivax, or Plasmodium falciparum. Its vector is female Anopheles mosquito. Now let us understand the life cycle. In mosquitoes, sexual cycle. When a female Anopheles mosquito bites an infected person, it ingests the parasite with the blood. The parasite undergoes sexual reproduction in the mosquito's gut, eventually forming infective stages called sporozoids that migrate to its salivary gland. In human, asexual cycle continues. First is the liver stage. When the mosquito bites a healthy person, the sporozoids enter the human bloodstream and travel to the liver. They multiply asexually inside the liver cells. Next is the blood stage. The parasite then bursts out the liver cells and enter the red blood cells, the RBCs. They multiply inside the RBCs, causing them to burst. The bursting of the RBCs releases hemozoin. Hemozoin causes the classic cycle of fever and chills. Why is it difficult to eradicate malaria? The parasite has a complex life cycle with multiple stages in two different hosts. It develops resistance to common antimalarial drugs. The mosquito vector is widespread, breeds easily, and can develop resistance to the insecticides. The parasite can remain dormant in the liver, as in the case of Plasmodium vivax, causing relapse months or years later. Chapter 8: Microbes in Human Welfare. Name two antibiotic producing microbes. Why are antibiotics ineffective against viruses? Antibiotics are the chemical substances produced by some microbes that can kill or inhibit the growth of other disease causing microbes. The two famous examples are Penicillium notatum fungus. They produce penicillin, and the second one is Streptomyces griseus. It is a bacteria that produces streptomycin. Antibiotics are ineffective against viruses because viruses do not have a definite cell structure. They lack their own metabolic machinery. Antibiotics typically target bacterial structure or functions like cell wall synthesis, as in penicillin, or protein synthesis, as in streptomycin. Viruses use the host cell's machinery to replicate, so antibiotics have no target to act upon. What is biogas? Explain its production in a biogas plant. Name the microbes involved. Biogas is a mixture of gases, primarily methane, along with carbon dioxide and hydrogen, produced by the microbial breakdown of organic matter like cow dung, plant wastes, etc., in the absence of oxygen, that is, under anaerobic conditions. Now, the production in a biogas plant. Slurry of organic waste is mixed with water and fed into the digester tank through the inlet. Inside the digester tank, anaerobic microbes, a group of bacteria called methanogens, example methanobacterium, act on the waste. They decompose the complex organic compounds, producing biogas, which rises and collects in the gas holder. The spent slurry is removed through the outlet and can be used as a rich fertilizer, the manure. The biogas can be piped out for use as a fuel for cooking, lighting, or generating electricity. Question 3. How do lactic acid bacteria convert milk into curd? Mention two additional benefits of LAB. When a small amount of pre-existing curd containing lactic acid bacteria or LAB like lactobacillus is added to milk, it is called inoculation. LAB multiply in the milk. They produce lactic acid, which causes the milk protein casein to coagulate and precipitate, converting the milk into curd. This process also partially digests the milk protein, making the curd easier for us to digest. Additional benefits of LAB: they improve the nutritional quality of food by increasing vitamin B12 content. They check the growth of the harmful disease causing microbes in our gut, contributing to gut health. Chapter 9: Biotechnology, Principles and Processes. Question 1. What are restriction enzymes? Why are they called molecular scissors? Explain their role in recombinant DNA technology. Restriction enzymes or restriction endonucleases are enzymes produced by bacteria that cut DNA at specific recognition sequences known as restriction sites. These sequences are typically palindromic, means reading the same forward and backward on both the strands. They are called molecular scissors because they precisely cut the long DNA molecule into smaller fragments. This is the first and most crucial step in recombinant DNA technology. Their role: they cut DNA at specific points, generating complementary sticky ends or the blunt ends. For example, the enzyme EcoRI cuts DNA at the sequence GAATTC, leaving short single-stranded overhangs. When DNA from two different sources, that is, human gene and a plasmid, are cut with the same restriction enzyme, they will have complementary sticky ends. These ends can then be joined together by another enzyme, DNA ligase, to form a recombinant DNA molecule. Describe PCR name the three steps and their purposes. Polymerase chain reaction PCR is a technique used to amplify, that means make millions or billions of copies of a specific segment of DNA. It is an automated in vitro process. It requires a DNA template, two primers, short single-stranded DNA sequence complementary to the target region, a thermostable DNA polymerase, that is Taq polymerase, and nucleotides. The three main steps in each cycle of PCR are: number one, denaturation. The reaction mixture is heated to a high temperature around 94 to 98 degrees Celsius. This breaks the hydrogen bonds between the two strands of the DNA template, separating them into a single strand. Second is annealing. The temperature is lowered around 50 to 65 degrees Celsius. This allows the primers to bind anneal to their complementary sequences on the single-stranded DNA template, flanking the target region. And the third process is extension. The temperature is raised to the optimal temperature for Taq polymerase, around 72 degrees Celsius. The enzyme adds nucleotide to the primers, synthesizing a new complementary strand for each template. This results in a double-stranded DNA molecule from the original one. These cycles are repeated 25 to 35 times, leading to exponential amplification of the target DNA. Now, question 3, draw label cloning vector PBR322. Mention functions of ORI, AMPR, and restriction sites. PBR322 is one of the first artificial cloning vectors developed from Escherichia coli. It is a circular plasmid DNA. Here are the key components and their functions. First one is ORI, or origin of replication. This is a sequence from where replication starts. It controls the copy number of the vector. A high copy number ORI allows many copies of the plasmid to be made within a single cell, which is essential for producing large quantities of the cloned gene. Chapter 10, Biotechnology and its applications. Question 1, what is Bt cotton? Explain how Bt toxin protects the plant. Bt cotton is a genetically modified, what is called GM crop. It contains a gene from the soil bacterium Bacillus thuringiensis, that is Bt. This gene codes for an insecticidal protein. Now let us understand the mechanism of protection. The Bt gene is incorporated into the cotton plant's genome. The plant cells now produce the Bt protein in an inactive crystalline form called protoxin. When an insect pest, such as the cotton bollworm, feeds on the plant, it ingests the protoxin. The alkaline environment of the insect's gut activates the protoxin, converted it into active toxic form. The active toxin binds to the specific receptor of the lining of the insect's gut's epithelium, creating pores in it. This causes cell lysis and swelling, disrupting the gut and leading to paralysis and eventually the death of the insect. Since the protein is only activated in the alkaline gut of the specific insect, it is harmless to the humans, other mammals, and beneficial insects that do not have such gut conditions. Transgenic animals are the animals that have had their DNA manipulated to possess and express an extra foreign gene. Their applications are vast. First, to study normal physiology and development. Transgenic animals are used to study the role of genes in normal body functions. For example, transgenic mice with an added growth hormone gene grew much larger than the normal ones, helping us to understand growth regulation. Second, to study disease. Transgenic models are created for human diseases like cancer, Alzheimer's, and cystic fibrosis. By studying these animals, we can understand how the disease develops and test potential treatments. For example, oncomice carry an activated oncogene and are used for cancer research. Third, for biological products, transgenic animals can be engineered to produce valuable proteins in their milk, blood, or urine. This is sometimes called farming. For example, Rosie, a transgenic cow, produced human protein enriched milk containing human alpha lactalbumin, which is more nutritious for human babies. Question 3. What is gene therapy? Explain SCID treatment using the gene therapy. Gene therapy is a collection of techniques used to correct a defective gene responsible for a disease. The goal is to introduce a functional normal gene into the patient cell to compensate for the non-functional gene. Treatment of severe combined immunodeficiency, that is SCID. It is a genetic disorder where children are born without a functioning immune system due to a defect in the gene for the enzyme adenosine deaminase ADA. Early approach, the first step was to isolate lymphocytes, a type of white blood cells from the patient's bone marrow. In vitro correction, using a retrovirus as a vector, a functional ADA gene was introduced into the lymphocytes. Reinfusion. The correlated lymphocytes were then reinfused into the patient's body. Outcome. This provided a temporary cure because lymphocytes are not immortal. The patient needed repeated infusions. Current approach, a more permanent solution is to isolate and correct the patient's own hematopoietic stem cells, which can produce all types of blood cells. When these corrected stem cells are reinfused, they are continuously producing a fresh supply of functional immune cells, providing a more permanent cure. Chapter 11, Organisms and Population. Define population and population density. Mention three methods to measure population density. A population is a group of individuals of the same species that live in a defined geographical area at a given time and have the capability of interbreeding. Population density, denoted as N, is the number of individuals of a species per unit area or volume at a given time. It is mathematically represented as N, the total number of individuals in a given region divided by area or volume. The three methods to measure population density are: first, the total count, directly counting all the individuals in the area, as for example, counting all plants in a small plot. Second, percent cover, For organisms that are numerous or difficult to count, like grasses in a field, we do not count each blade. Instead, we estimate the percentage of ground covered by that species. And the third method is indirect method, pug mark or tracks. For elusive or wide ranging animals like tigers in a forest, we count their indirect signs, such as pug marks, the footprints, or fecal pallets, to estimate population size. Question 2. Explain any two: predation, competition, parasitism, mutualism with example. These are important interspecific interactions where the population of one species affects the population of another. The first one is predation, an interaction where one organism, the predator, kills and eats another organism, the prey. Example, a lion, predator hunting and killing a deer, prey. Predation helps in transferring energy up the food chain and keeps prey population in check. Second is competition, an interaction between two species or individuals where both are harmed as they compete for the same limited resource, such as food, space, or mates. Example, when two species of birds, that is, the two species of finches, compete for the same type of seed on an island. This can lead to competitive exclusion or resources pardoning. Parasitism, an interaction where one organism is the parasite which lives in or outside the body of another organism, the host, from which it derives nutrients causing harm to the host. Example, a tick parasite living on a dog host, feeding on its blood. The tick benefits, the dog is harmed. Fourth one is mutualism, an interaction where both the species involved benefit from the relationship. Example, lichen, a mutualistic association between a fungus and an algae. The algae provides food through photosynthesis, and fungus provides shelter and absorbs water and minerals for the alga. Another example is mycorrhiza, the association between the fungi on the plant root or the nitrogen fixing bacteria in the root nodules of the leguminous plants. Question 3. What are xerophytes? Mention two adaptations in desert plants. Xerophytes are the plants that are adapted to survive in dry, arid environment like arid deserts where water is scarce. They have evolved various morphological and physiological adaptations to conserve water. Chapter 12, Ecosystem. Question 1. Define ecosystem. Explain its biotic and abiotic components. An ecosystem is a functional unit of nature where living organisms like biotic community interact among themselves and with their non-living abiotic physical and chemical environment. It can be of any size, from a small pond to a large forest. An ecosystem is composed of two main interacting components. First is abiotic component or the non-living component. These are the physical and chemical factors of the environment. Physical factors like sunlight, temperature, rainfall, humidity, and the soil structure. Chemical factors like the availability of essential inorganic substances like carbon, nitrogen, phosphorus, oxygen, water, and various minerals. Biotic components, the living ones, these include all living organisms which are classified on the basis of their mode of nutrition into producers, autotrophs, organisms like green plants and algae that can produce their own food through photosynthesis. They form the basis of any ecosystem. Second group is of the consumers or the heterotrophs, organisms that cannot produce their own food and depend on the producers. They include herbivores, the primary consumers, carnivores, the secondary consumers, and the omnivores. And the third group is of the decomposers, saprotrophs. Organisms like bacteria and fungi, they break down dead organic matter, detritus, into simple inorganic substances, returning them to the soil to be reused by the producers. They play a very, very important role. Second question, explain the 10% law of energy transfer. Calculate energy available to tertiary consumer if producers have 10,000 joule of energy. The 10% law was proposed by Lindemann. It states that only about 10% of the energy available at one tropic level is transferred to the next higher tropic level. The rest of the energy, about 90%, is used up in the metabolic processes like respiration, growth, reproduction, etc., or is lost as heat to the environment. Energy transfer to the tertiary consumer T4, 10% of 100 joule, that is 10 joule. So the energy available to the tertiary consumer snake is only 10 joule. This explains why food chains rarely have more than 4 to 5 tropic levels. This is simply not enough energy left to support another level. Chapter 13, Biodiversity and Conservation. Question 1. List 3 causes of biodiversity loss. Explain habitat loss or fragmentation. Biodiversity loss is a major concern. The 4 main causes are often summarized as the evil quartet: habitat loss and fragmentation, overexploitation, that is, overfishing or hunting, alien species invasion, that is, introduction of exotic species, co-extinctions. When one species goes extinct, it can cause the extinction of another species dependent on it. Explanation of the habitat loss and fragmentation. This is the most important cause of biodiversity loss. Habitat loss. When humans clear forests for agriculture, urban development, or dam construction, the natural homes of countless species are destroyed. Species that cannot adapt are forced to move or die. Fragmentation. Sometimes a large continuous habitat is divided into smaller isolated patches, that is, the fragments, by roads, railways, or the canals. These fragments are often too small to support viable populations. The edges of the fragments create new microclimates, and animals in small, isolated fragments may not be able to find mates or food, leading to inbreeding and local extinction. Second question, differentiate between in situ and ex situ conservation with examples. Conservation strategies are broadly classified into 2 categories: in situ conservation and ex situ conservation. In situ means on-site conservation, protecting an endangered species in its natural habitat. Its goal is to protect the entire ecosystem, allowing species to evolve and function naturally. Some examples: construction of the national parks like Kaziranga for rhinos, wildlife sanctuaries, biosphere reserves, sacred groves. Whereas ex situ conservation means off-site conservation, protecting an endangered species by removing part of the population and placing it in a protected artificial setting. This is to ensure the survival of an individual species, often with the goal of reintroduction. The examples of it are botanical gardens, zoos, seed banks, gene banks, aquariums. Question 3. What are biodiversity hotspots? Why are they important? Biodiversity hotspots are regions on Earth that are both exceptionally rich in biodiversity, especially endemic species, species found nowhere else, and under serious threat of destruction from human activities. To qualify as a hotspot, a region must meet two strict criteria. First is it must have at least 1500 species of vascular plants as endemic. And second, it must have lost at least 70% of its original habitat. Why are they important? They represent areas of high biological value and conservation priority. By focusing conservation efforts on these hotspots, which cover a small percentage of the Earth's land's area, we can protect a disproportionately large number of species, over 40% of plants and vertebrate species. Examples of hotspots include the Western Ghats and the Himalayas in Examples of hotspots include the Western Ghats and the Himalayas in India, which harbor thousands of endemic species facing severe threats. Students, we have successfully completed our comprehensive journey through the entire Class 12 biology syllabus. We have covered the most important exam relevant questions from every chapter, explained each concept in detail with example and diagrams. I hope this detailed walkthrough has not only clarified your concepts but also boosted your confidence for the upcoming exams. Remember, consistent revision, practicing diagram based questions are the keys to success. Study smart, stay healthy, and believe in yourself. All the very best. Thank you, students, for watching Asha Bansal's